This is the beginning of the Chapter 4 lecture on tissues, the living fabric, beginning with PowerPoint 4A, Slide 1. In this chapter, we will be exploring the different tissues. Tissues differ from each other in the types of cells. They also differ in the amount of materials between the cells and the kinds of functions that they perform to help maintain the body. So that's what we'll basically be talking about, defining the different tissues and understanding those characteristics. The four types of tissues that we will study include epithelial, which is a covering, connective, which is for support and connection, muscle tissue for movement, and nervous tissue, or nerve tissue, a conducting tissue. Let's begin with epithelial tissue. Notice its characteristics. Epithelial tissue is almost entirely composed of cells. There's very little extracellular material. Cells are tightly packed together and joined by structures like tight junctions and desmosomes. Epithelial tissue also comes in sheets. Cells that are located at the top are referred to as the apical cells. So the top surface would be the apical or superficial surface, and the bottom is the basal surface. This gives epithelial tissue the characteristic of polarity. Epithelial tissue is always glued to a layer of connective tissue, and it's glued by an acellular material composed of glycoproteins we call the basal lamina. The basal lamina glues the epithelial tissue to the first layer of connective tissue, known as reticular lamina. And together these layers, the reticular lamina and the basal lamina, make up what we refer to as the basement membrane. Epithelial tissue is avascular, meaning that it contains no blood vessels. It is innervated. It is supplied by nerve fibers. Epithelial tissue also has regenerative capabilities. It can replace lost cells by cell division. We classify epithelia based on the shape of the cells, as well as how many layers are contained within the sheet. If there's only one layer, it's referred to as simple. If there are multiple layers, it is known as stratified. The different cell shapes include squamous, cuboidal, and columnar. Squamous cells are flattened cells. Cuboidal cells are cube-like, and columnar are tall, column-like cells. Together, the cell shape and the number of layers will indicate what type of epithelial tissue it is. For example, simple squamous is one layer of flattened cells. Let's begin with simple squamous. As the name implies, this is a single layer of flattened cells. Functions include diffusion and filtration. It also provides a slick, friction-reducing lining. It's present in the kidney glomeruli, in the lining of the heart and blood vessels, the lymphatic vessels, and the serosae. In this slide, you can see a micrograph of simple squamous epithelium. This particular sample is part of the alveolar or the air sac walls in the lungs. Again, simple squamous epithelia is a single layer of flattened cells. It allows for materials to diffuse through or to be filtered through. It provides a frictionless surface, and it's located in places like the kidney, glomeruli, the air sacs of the lungs, lining of the heart, blood vessels, and lymphatic vessels. It also makes up the lining of the ventral body cavity we refer to as the serosae. The second type of epithelial tissue is simple cuboidal. Again, this is one layer of cells. However, the cell's shape will be cube-like. Nuclei will be spherical. Functions include secretion and absorption. Simple cuboidal epithelia is present in kidney tubules, in the ducts and secretory portions of glands, and ovary surfaces. In this figure, you can see a micrograph of simple cuboidal epithelia. Simple cuboidal epithelia is a single layer of cube-like cells. The function includes secretion and absorption, and is found in kidney tubules, ducts and secretory portions of small glands, and the ovary surface. Another type of epithelia includes simple columnar. This is a single layer of tall or column-like cells. Oftentimes you'll see the cells containing cilia, those little hair-like structures that create a current so substances can pass across the cell. Goblet cells are unicellular glands that are often found in a simple columnar layer. Functions of simple columnar epithelia include absorption and secretion. The non-ciliated type are found in the digestive tract lining and the gallbladder. The ciliated type are found in the small bronchi, uterine tubes, and some regions of the uterus. Notice the structure of simple columnar epithelium. We have a single layer of tall column-like cells. In this figure, you can actually see an example of a goblet cell releasing its mucin or mucus into the lumen area, the opening. Functions of the simple columnar epithelia include absorption, secretion of mucus, which is indicative of those areas where goblet cells are found. Locations include, for example, non-ciliated types in the digestive tract, and the ciliated type in small bronchi, uterine tubes, and other regions of the uterus. Another type of epithelial tissue is pseudostratified columnar. This includes cells that are column-like, but the cells are at different heights in regard to the apical surface, so it appears as though the nuclei are at different levels, giving a false appearance of many levels. However, it is a single layer. Pseudo means false. Stratified means many layers. So the word literally means false, many layers. So it is really only one cell layer thick. Function of pseudostratified columnar includes secretion and propulsion of mucus. So we also see goblet cells within this type of epithelia. Location include male sperm carrying ducts, which are non-ciliated, and the ciliated type is found in the trachea. This is a micrograph of pseudostratified columnar. It's a single layer of cells of differing heights. Some reach the apical surface. The nuclei are seen at different levels. It gives it the false appearance of being stratified. However, it is only one cell layer thick. Like simple columnar, pseudostratified columnar also contains goblet cells and may be ciliated. Functions include secretion, particularly of mucus, propulsion of mucus, specifically by ciliary action. Non-ciliated types are found in male sperm-carrying ducts and ducts of large glands. Ciliated varieties line the trachea and most of the upper respiratory tract. Stratified squamous contains many layers of cells that are flattened, and because there are many layers, this type of tissue functions more in protecting areas that are subjected to abrasion. So if we think about the areas that are subjected to abrasion, it helps us to understand the location. Your skin, for example, contains keratinized stratified squamous epithelia. The lining of your mouth contains non-keratinized stratified squamous epithelia. In this slide, you can see a micrograph of stratified squamous epithelia. It's composed of many cell layers. Basal cells often appear cuboidal or columnar. Surface cells are flattened and squamous. So the apical surface, because it is squamous, gives it the term stratified squamous. There are keratinized examples and non-keratinized examples of stratified squamous. The function of this epithelial tissue is to protect underlying tissues that may be subjected to abrasion. It's found in areas such as the skin, which is keratinized, and other non-keratinized types are found in the lining of the mouth, esophagus, and vagina. Stratified cuboidal and columnar cells are rare. Stratified cuboidal epithelia is found in some sweat glands and mammary glands. It's typically only about 2 cell layers thick. Stratified columnar also has a limited distribution. It's found in the pharynx, the male urethra, and other glandular ducts. It also occurs at a transition area between 2 types of epithelia. Another type of epithelial tissue to examine is transitional. It is several cell layers thick, and usually at the basal layer, the cells are more cuboidal and sometimes columnar. At the apical surface, the cells become more dome-shaped. The function of transitional epithelial tissue is to provide stretch. So the location of this type of tissue is indicative of areas in the body where we need stretch. For instance, in the urinary bladder, when it fills with urine, it needs to be able to stretch to accommodate the urine, as well as the tubes that drain from the urinary bladder or into the urinary bladder, the ureters and the urethra. In this micrograph, you can see transitional epithelia. At the basal layer, the cells are often cuboidal or columnar-like. At the apical surface, they appear to be more dome-shaped and almost appear to be bubbling out. The function of transitional epithelia includes stretch, so it provides stretching. It's found in areas such as the urinary bladder, the ureters, and part of the urethra. The types of epithelia we have covered so far are for lining and covering. In this slide, you see that epithelial tissue is also responsible for forming glands. Glands are classified based on two things: the site of product release and also the number of cells forming the gland. Glands that secrete their product directly into the extracellular space are known as endocrine glands. They're often referred to as ductless glands. Endocrine glands secrete hormones, amino acids, proteins, glycoproteins, and steroids. Exocrine glands secrete their products onto the body surface or into a cavity. Exocrine glands secrete substances like mucus or sweat. Those are known as sudoriferous glands, or oil, known as sebaceous glands, and saliva, known as salivary glands. One important unicellular gland is the goblet cell. It's found in simple columnar and pseudostratified columnar epithelial layers. Multicellular exocrine glands are composed of a duct and a secretory unit. Exocrine glands are more numerous than endocrine glands. In this slide, you see a goblet cell. A goblet cell is a unicellular exocrine gland, typically found in simple columnar and pseudostratified columnar epithelia. The goblet cell looks like a cup or a goblet. It contains many vesicles of mucin. The mucin will dissolve in water to form mucus, which helps to protect and lubricate surfaces. The Golgi apparatus is a very important organelle to goblet cells, in that it is needed to package the mucin contained in the secretory vesicles. Multicellular exocrine glands are classified according to the duct type, which is either simple, meaning it does not branch, or compound, meaning that the duct does branch, and also the shape of the secretory unit. In this slide, we see structural classification of multicellular exocrine glands that have a simple duct structure, meaning that the duct does not branch. If the secretory unit forms a tube, it's referred to as tubular. If the secretory unit forms a balloon or sac-like structure, this is known as alveolar. Again, what we're noticing is the duct area is not branching on any of these. This slide shows you an example of multicellular exocrine glands that are compound, meaning the ducts branch. So look at the brown structure. They're very branching. Again, if the secretory unit forms a tube, it's referred to as tubular. If the secretory unit forms something that looks like a balloon, it's alveolar. And if it's kind of in between a tube and a balloon, it's referred to as tubulo-alveolar. Multicellular exocrine glands are also put into groups based on how their products are secreted. In Murocrine glands, the products are secreted by exocytosis. Murocrine glands are found in the pancreas. They're sweat glands or sudoriferous glands and salivary glands. Holocrine glands secrete their products when they rupture. The whole cell must rupture. A good example would be the sebaceous glands or the oil glands. This figure shows the different modes of secretion in human exocrine glands. A represents murocrine glands that secrete their products by exocytosis. B represents holocrine glands. The entire secretory cell ruptures, releasing secretions and dead cell fragments. A third type of multicellular exocrine gland that should be mentioned are apocrine glands. Apocrine glands accumulate their products, but they only pinch off from the surface, so the whole cell doesn't rupture. There's controversy if there are even apocrine glands in the human body.